you have two ropes, one connected to the wall, one on the other side. These two ropes come like this, and they have some directionality. Maybe somebody might have covered this, but think about that you are trying to walk on the first rope, and you have a glue on your feet, and you're trying to walk on that rope, and at the same time you're trying to move with respect to the other rope. And what ends up happening is both of these ropes get taut. So that's how this force is being generated. So this is the amount of each force generating motors here is on the order of piconewtons, 2 piconewtons. Okay? So every time the motor wants to walk forward, and if you hold the rope, then this is the amount of force it will generate. So every time it tries to walk, but then it cannot walk because you're holding the rope, it is generating the force, and the amount of force it generating is shown by these peaks. So these peaks are about 2 piconewtons. So this is the, so if there are many, many motors involved, then many of them are trying to move, and the overall force can be significantly higher. So in a focal adhesion complex, there could be hundreds of these motors, so you can generate force on the order of hundreds of piconewtons. Okay. Hundreds of piconewtons. So that gives you an idea. So it's about maybe 0.1 nanometer, 0.1 maybe 0.05 nanometer. Okay. And if you have a focal adhesion complex with hundreds of these ropes that are connecting to the rest of the cell, you can generate force on the order of a nanometer. Okay. Now, if you have many, many complexes, then your force could be on the order of tens of nanometers. So that's our baseline. That's our baseline. So now imagine you put a cell on a substrate, and what does the cell do? Initially, it has no anchorage yet. It comes down on a substrate, and it begins to form those transmembrane molecules, integrins coming out. Right? It begins to form this anchorage. While it is beginning to form this anchorage, it's also beginning to build a force. Right? Because that's where these motors are getting activated. Right? So imagine first anchorage comes on the wall, other anchorage comes on the other wall, so two ends of the cell are just forming. This is the early stage. And these motors are getting activated, and the force is beginning to build up. So in your mind, you can imagine at the early phase, you may have on the order of tens of piconewton forces that the cell is generating. And as time goes by, maybe by about 10 to 15 hours, all the motors have been engaged. The cell is fully stabilized. The force builds up to on the order of tens of piconewtons.
What kind of cells are those?
This is the one that in the picture?
Yeah.
These are fibroblasts. I must have somewhere. These are kidney fibroblasts, multi-kidney fibroblasts. This one and both of these two.
What is the source of this force?
The source, as I just mentioned, are the molecular motors. Did anybody cover molecular motors?
I think Scott mentioned that.
Scott mentioned that, right? Okay. So the molecular motors' job is you can imagine they are like walking machines. So their job.
She was not here first day.
Okay. But in short, and this is a whole topic by itself, but in short, these molecular motors are I don't know, I was not prepared to talk about molecular motors, but maybe I can describe very briefly. So they are like molecules which has two arms. They grab at 2 points of a rope. Just imagine the rope being, in this case, the actin filament. So they are like grabbing the rope. So they grab the rope, and then the other arm is about to grab the other point. So when this happens, then this wants to move, while the other one wants to go to the next one. So if the rope is free, then you would see the rope is just sliding. Okay? So there are cool videos that you can download from the web. So what people do is they take those fibers, the actin filament, and attach the molecular motors on a glass substrate. Okay? So it's like molecular motors, 2 feet are attached, glued to the substrate, and then on top there are lots of actin fibers going. So what would you expect from such an experiment? So actin fibers are floating around, and they can grab it. So molecular motors, the 2 attached, glued to the substrate. The other two hands are free. So what do you expect? The only job of these molecular motors is they grab and then take the next one. And then grab, just move. So what do you expect from an experiment?
The fibers would you see the fibers move around.
Yes,
And then
that's right. You will see the fibers moving all the time. So, and you can take a simple microscope, just see the fibers are just moving. So every time they get grabbed by a motor, then you can see the fibers are moving. And from the speed, you know how fast these motors are working on the fibers. But there's an energy associated with it, right? Nothing happens without, nothing can move without cost of energy. So where do you think the energy should come from? ATP. So the motor's job is to consume an ATP, produce an ADP through phosphorylation, and some energy is associated with it. And so that's why every time you see a peak in the motor's motion, every time you see a peak, that means an ATP has been consumed, right? And it takes a step. It goes from one jump, one grab to the other. So there's work done, and ATP has been consumed. So, in an experiment that I just described, where you see the motors are moving around, so if the ATP is very, very low, if you provide just a little bit of food, then you will see once in a while these motors are activated, right? Because there's not enough ATP. Yes?
It seems the force is very small. How to figure out is the noise signal or is a real signal for your?
Oh, the way these experiments are done are, so you can attach a bead to one of these fibers, for example, and then you can come in and hold the bead. And that prevents the fiber from moving, and you measure the force that you need to hold the bead, that gives you the force. So basically, each force peak is because the bead is preventing the fiber from motion, and they are holding from moving.
Have you measured those forces in different cell types, like cardiomyocytes?
No, these are in vitro studies. So
yeah, but in vitro.
Yeah, in vitro means you take the motors out of the cell.
Oh, the motors.
That's right.
You're not inside the cell.
No, I don't think there has been any measurement inside the cell in vivo. Because first of all, there are many different types of motors involved. So these are in vitro studies. And in fact, only recently, I would say over the last 10 years, people have begun to understand the activities of the motors. In fact, there is still debate precisely how they act. There are debates in terms of is the motion like walking motion, or is there a kink in motion? There's a lot of literature on this one. But the essence is they grab, and then the next grabbing happens, ATP is consumed, the force gets generated. It wants to walk. If you don't let it walk, it generates the force.
So what's like till when will it generate this force? What happens beyond the point? Like it's obviously that force that's being generated when it cannot pull at that
Right.
is creating a tension.
Mhm.
So eventually either the filament has to snap or something has to happen with the protein. So
Yeah.
what's the snap? Is there any idea what that snapping point is?
Yeah. So if you hold the bead, right? So no motion is going on. So every time it's trying to walk, that's the maximum force you measure. That gives you an idea how much is the capacity, what's the force generation capacity of the force. In vivo, when it is inside the cell, and I'll give you a specific example. In vivo inside the cell, when the forces are being generated, so the motors come in and go. And this is a little bit statistical mechanics concept. So the motors is not permanent. So I think about two fibers. If I put one motor there, it's going to try to build the force, and then it would be staying there, and that would be the maximum force, right? But what happens is there is a certain probability this motor will get off. Okay? So let's say this probability is 10%. So when the motor grabs this fiber, there is a 90% probability is get off. So then the force will go up and then go to 0. But in the cell, the force is always being produced. So how is that possible? How do you think it is possible?
Many more motors.
There are many, many motors. Some are running, some are living, right? So on an average, there is a certain number of, mean number of motors that are always there. So that's how. Now, as I said, it's a specific example. So every time our heart beats, okay, that's the same process. So that's a specific in vivo example. So many motors, they basically in each cell, each cardiac cell, many motors get activated and pull the actin fibers that contracts the cell and the whole heart beats. Okay? Now the question is, how do you have relaxation? Why the heart goes back? Right? What could be the possible mechanism the heart goes back? What's that? Depolymerization of the fibers? No, because if you depolymerize, then you have to repolymerize.
Either
Polarization? No, it's a very simple idea.
are the motors reversible? Can they go in both directions?
No, the motors are asymmetric, so is the fibers.
Okay.
Right? That's why directionality comes in. You see, once in a while, there's a negative force. Just once in a while. So that is because that there is a lower activation barrier walking forward than walking backward. So there is much less probability of walking backward. So what happens in the heart is, it's a very simple idea. So the motors are activated, so they pull the fibers, your heart beats, right? It contracts because the force is being built up. Right? But you don't want the heart to be always contracted, right? Because then how do you get the next cycle, right? So there has to be a way to release it, right? And so what do you need? You need two things to release. One is the force has to go down to zero. And there has to be some elasticity so that the heart can go back. So I can take a spring and compress it, but if the spring is kind of broken, it's a chewing gum, it's not going to help if I take the force out. Right? So you need the elasticity of the heart so it can go back by itself. Right? So that's when it recaptures the blood again, right? But the mechanism by which the force is released is very simple. Is that, think about this rope. I'm pulling this rope. Now, one way I can disengage them is if I make the rope very lubricated. If I cannot grab anymore, right? So if the motor wants to bind it, but if something else is there, which is kind of lubricated, I cannot grab it anymore. Then the force is back to zero, right? So what happens is there is a complex called troponin 1 that always coats these fibers, actin fibers. So it is a long story, but the bottom line is every time the heart beats, the troponin gets off, the actin gets bricked by the motor and pulls it. And then when the pulling job is done, troponin has to go back. Even if it is holding it, it has to give it up. So the troponin lubricates the fiber and it goes back. So every time our heart is beating, this calcium signal goes in that makes the troponin get off and get back, get off and get back. So when there's a heart attack, the first thing we should check is in the blood, is there troponin or not? Because in case of a heart attack, the troponin which is supposed to go back does not. So the motors have a permanent grip on the actin. Where does the troponin go? It goes to the blood. So the first thing the doctor checks is what is the level of troponin. If troponin level goes up, that means you had a heart attack. So I was not planning on this one, so I'm not so sure how much I can cover. Any other question? How the force generates? That's intracellular force. Okay, so we need to measure this force. So what happens with it? You must have covered, maybe Jimmy covered this already. The differentiation. Okay, so how do we measure the force? Okay, so there are multiple ways of measuring the force. Some came from material science, some came from motivated by biology. So atomic force microscope. How many of you heard atomic force microscope? Haven't heard atomic force microscope?
Yes, I thought that
Atomic force? Oh no. But I don't expect you have worked with atomic force microscope. But any idea about how it works? No? Atomic force microscope. So it's a very simple idea. It's a cantilever. You have a simple cantilever, and if you dive, on a diving board, when you stand on, the diving board goes down. If the heavier the weight is, the more it goes down. That's simple idea. So the same thing that on the force microscope is that this is a simple cantilever. It contacts some point, and if you apply the force, then the cantilever bends, right? Cantilever bends, and the amount of bending gives you the amount of force. So that's atomic force microscope. The second is optical tweezer. So what it does is, if you shine a laser beam on a bead, then the laser beam produces a force on the bead. So if you want to take the bead away from the laser where it is shining, then the laser force brings the bead inside. So anybody talked about optical tweezer in this session? No?
Not in detail, but someone covered.
Okay, okay. So you have heard a little bit. So optical tweezer.
I talked about the Cellular CPD.
Okay, good. So optical tweezer can measure also forces on the order of 200 piconewton. It's very hard to go too much force because you can imagine the light force is not that much. So when you shine the light, there is some force on your body. Did you feel the force when you turn on the light? It's radiation force, very, very small. So optical tweezer force is always not a radiation force, but it's related to the way the light gets through the bead. So it generates higher force than atomic force microscope. And then you can have a magnetic bead where you can put the beads on a cell, and then you can generate force on the magnetic beads. Or if the cell is producing the force, you can hold the bead and get a measure of the force. Okay. So these are the ranges of the force. Atomic force microscope has been there for quite some time. Optical tweezer is relatively new, maybe on the order of 10, 15 years. Magnetic bead has been there for much longer time. So people have been using these tools to measure the cell forces. So what do you see as a limitation in measuring the force other than the maximum forces that you can measure? So now you have an idea about the force range, right? Each motor is producing an order of a piconewton force. An order of piconewton force. There may be hundreds of them involved. So the force range is on the order of tens of piconewtons to tens of nanonewtons. That's the range you want to capture. So obviously, because of the lower range, it's hard to get to a nanonewton by the force microscope or optical tweezer. So your force range is not covered in a single cell measurement. Magnetic bead probably goes as high as 10 nanonewton, but sometimes the cell can produce the order of tens of nanonewtons. So again, you're limited to force range. Is there any other limitation that you see when you look at the image? So you put the optical bead sort of randomly on the cell and then stick to the cell. So is the magnetic beads sitting on the cell, falling randomly on the cell. Okay? So do you see a limitation in these measurements?
It need not be right on top of that force, right? So you would be getting it at an angle, so you get something lower.
Right. You cannot control where to get there. Now, with the optical bead, there's a little bit better control because you can capture the bead and then begin to move around, but it becomes more complicated. So if you want to measure the force at the right angle at site, it's very difficult to take it to the right place. Right? So these are the limitations. There is another approach, which is also relatively new, maybe 10 years or so. It goes back to the very basic mechanics. Here, you can put a cell, culture a cell on a substrate which is soft, an elastic material, soft, and the cell produces the force, so the substrate begins to deform, right? And if you know how much the substrate is deforming, then you can back calculate the force. It's like the idea of a spring when you stand on the weighing machine, so the spring deforms a little bit, right? And the weighing machine measures how much of the spring deformation that gives you the force. The same idea here. It's not a spring, but the idea is the same. It's a 3D material, and you put some beads that are fluorescently labeled. So inside the bead are these many beads, and when the cell produces the force, the beads move. They themselves not move with respect to the gel on which the cell is sitting, but the whole gel deforms, right? And hence the beads begin to move with the gel. So these beads are just points where the spring deformation is measured, and you can compare if the bead was here before and now the bead is over here, so you can put the location of the beads in a computer program that calculates from using finite element, which I suppose Jimmy might have covered a little bit, that gives you the overall force field. So that tells you how much the force the cell is generating. So what you get is after you see where the beads are from the bead motion, you can find out how much they have moved. So this is the cell which is producing forces over here and here. It's kind of pulling the bead this way, pulling the bead the other way, and you can from this motion of the beads you can back calculate the force that the cell has generated. So, but here in this case, wherever the cell goes, whatever the force it generates, that's the measure. You don't go and say, well, I want to find out, what's going on at this point on let's say somebody near the nucleus. You don't get a specific local force field. You just get an overall force field. And there are other limitations that are involved. Okay, so skip this one. So based on what you have seen so far is simple ideas. The force that the cells generate is on the order of hundreds of piconewtons to maybe tens of nanometers. The atomic force microscope, optical prism, magnetic bead, they can measure force from few piconewtons to on the order of 10 nanometers. The range is not that high. The other problem is if you want to measure 5 inches length, your scale probably would be very, very fine. You want to measure, let's say, 0.1 inch accuracy. Right? But if I tell you that can you measure 10 miles distance, would you like to use the same scale? Obviously not, right? Because you don't need that accuracy. So the measurement technique depends on the range of measurement. So the longer the range, you give up the accuracy and measure, use the higher scale. But in the case of cell, the cell at the beginning wants to generate small amount of force. By the time it ends its cycle, it's generating tens of nanonewtons. You want to have some way to measure the force which is very, very small to very, very large. None of these techniques can do that. Right? Atomic force microscope maximum range is 50 piconewton. It can measure 1, 2 piconewton very precisely. But it cannot measure 10 nanonewtons because it's way outside the range. Just like you would use a small scale, fine scale to measure 1 inch or 10 inches, you will not use the same scale to measure a mile. Okay? We're talking about a piconewton is 1,000 times less than a nanonewton. We're talking about 5 orders of magnitude force. How do we solve this problem? So one way would be to design something as like a spring, and in a spring, if you coat the spring with some kind of material that is functionalization, like extracellular matrix, you can bring this in contact with the cell, and the cell would generate, as I mentioned before, the integrine binding with the extracellular matrix so that you create a physiological anchorage that I mentioned before, physiological anchorage. And then if you pull it, then this spring is going to deform, and from the deformation of the spring you can find out what is the kind of force that the cell is generating. Or you don't have to pull it, you could just leave it there, and the cell may begin to pull, and then you can measure the force by looking at the spring deformation. Okay? Simple idea. So one realization of this would be, if you think of some microscale beams attached to some rigid component, and if you bring in contact with the cell, and the cell may begin to pull this, and you can find out what is the total deformation of this beam, that will give you the force of these beams, which means the force the cell is producing. Okay? It's the same idea as the atomic force microscope, but in a slightly larger scale. Now, the question is, if you want to measure force that are very small at the same time very, very large, so what do you need? Yes?
For the cell, when you want to measure the pole of the cell, do you consider the substrate force when you put the cell, or you make system?
It's an independent. It doesn't matter, because if the cell is sitting on a substrate, and then you bring in the probe and bring in contact with the cell, then the probe feels the force that it is subjected to. The cell produces the force, and that's the only force it measures. Right? There could be other sources of force, but it's not measuring the whole thing. It's measuring the point contact force directly. So one way to measure the small force to large force, and this is a concept I want all of you to take with you at home. Okay? So one way to measure small to large force is to have a sensor that is very, very soft, that can measure very fine force. So what do I mean? How do we measure very fine force? So a way to measure the force is you measure the deflection of a cantilever, the deflection of something in a spring. And if it deflects in a measurable way by a small force, then you measure the force with high resolution, right? With high resolution, because you can find how much it is moving, right? By a small force. Now, if you apply a large force, then if your spring continues to deform by a large amount in a linear way, then you are done, right? Then you are done. So if you have an atomic force microscope, a cantilever, that deforms and you measure this deformation because of a very, very small force. But if the force is very large, then it keeps deforming. And you calculate the force because of the deformation. That's all it means. As long as there is an output related to the force, you are fine. So why does it not work with atomic force microscope? Yes?
Cantilever material cannot be as sensitive to a large and a small.
Right. So you cannot keep on deforming. It's going to break, or the deformation will not be linear anymore as a function of force. You want, if you double the force, you want the doubling the deflection. At least you would like to. You don't have to. So something that can measure small force, if you apply too much force, it's going to bend, break. Too much deformation. Deformation goes way out of the linear range and so on. If you keep that linearity, then you are fine. So, in other words, if you can keep the linearity, then you are fine. So one way to keep the linearity and measure the small force is to have a series of these kind of springs. So this is like one demonstration, a series of these springs. So what does it do? How does it achieve the high resolution and large force? So what happens is if you apply a small force at the top, so the spring constant of a series of springs, so if I have spring attached to it, and then another spring attached to it, another spring attached to it, each one is K. What is the resultant spring constant? What's that? 3K? 130. Oh, big difference. Okay, so how do you calculate the spring constant? What is this? So this is springs in series, right?
Yeah.
In contrast to a case where you have a spring, another spring, another spring. So again, K, K, K. And if you apply a force here versus if you apply a force here. So how do you compare? So in this case, because each spring is holding it back, right? So in this case, the spring constant is 3K, right? You just add it up. So basically, you do K plus K plus K, right? In this case, the spring constant is 1 over K plus 1 over K plus 1 over K is 1 over, let's say, K star, which is the resultant spring constant. Right? Does it make sense? So this would be 3 over K, and then K star is K over K. Right? So now if you take a spring that you can make with spring constant K, so higher this number, if this number is, let's call it N, and K star is N over so K over N, right? So I'm going to have a quiz at the end of this class. So K over N, so if I can make N very large, then my effective spin constant goes very, very small. Right? I made a very soft spring. What that means is the deflection, if I apply a force F, F is K star times displacement, right? K star times delta. So if I measure a delta over here, due to a force F, then the force F is K star times delta. So if I can measure delta, if my K star is very, very small, then I can measure very small force. Let's say I can measure only 1 micron displacement by looking at the optical microscope. So if my K star is very, very small, my force measurement can be very, very small. Okay? But the trick is I want small force, but I also want very large force by the same tool. Right? So what happens is, in this case, physically, you can imagine, when I have a delta displacement, how much is the displacement in each spring? How much each spring is deforming?
A third of it.
A third of it, right? So each spring is deforming a tiny amount, but the net displacement is large. Now, if I can measure, so if I have only one spring, then the force would be F equals K times the, let's call it delta 1. Okay? So if K is large, okay, K is large, then I can measure large force by measuring corresponding delta 1. Right? So if I now apply this large force, let's call it F large. If I apply this F large over here, what's going to happen? The whole thing will have a huge displacement, right? But each spring is going to be still linear because F large is this guy, F times delta 1, which is still linear. So each single spring still linearly stretches, but the net total displacement is very large. So I can still measure this F large. The only thing is my net displacement delta, which would be the sum of F delta 1, delta 2, and so on, would be large. So I still can have large force measurement by adding the series of springs. So here, I have many of these springs. Each one is deforming like this one. So the force goes from this spring to the next, to the next, to the next. Here, when it is in parallel, they don't go from one to the next. Each one is equally active. Here, each one is also active, but the force goes from one spring to the next to the next. That's in series. So here, when I apply the force at the end, the first spring deforms, but then transfers the force to the next, and then the next transfers to the second one, and so on. They are getting transferred one after the other. So there's springs in series.
So what exactly is the way by which you measure displacement?
What's that?
What is the way you measure the displacement?
Just by looking at it. See, X is a displacement.
No, I'm saying in practice.
In practice, optical microscope.
So an optical microscope that can measure really tiny displacements as well as.
Tiny means in optical microscope you can easily get on the order of 0.1 micron. In fact, you can improve the resolution by some other techniques, but you can easily get 0.1. And of course, you can measure tens of microns. So that's the whole idea, right? Yes? Yeah.
In each pair of arms, their length is a little bit different. So when we're interested.
No, each of them I did not draw to scale here. Right? So each length, this one would be maybe slightly larger, just to hold it. But the rest of them would have the same length, except the last one would have half the length.
Okay, so in the middle, big chunk of the middle section.
Big chunk would have many of these springs, right? So that's one way. Now, this is basic. There's nothing to do with microsystem. This is true for any system. The springs in series is a fundamental constant. Right? So this is true for large, small systems, doesn't matter. Does it make sense? Any other question? So the idea is to put the springs in series so the resultant spring constant becomes very small because it's k over n, the number of springs attached to it. So that gives you the lower spring constant. So you have the small spring constant and you have total force displacement can be measured. So how can you have this kind of springs? You can put make these springs and put it in a chip, and the chip will be held by an actuator, like a piezo actuator that moves X, Y, Z, and you can bring the tip of this sensor in contact with the cell, and you can measure the force on the cell. Okay? So the physical realization is very simple. So basically you take the same thing, and the piezo actuator just hold this part, makes in contact with the cell, the tip comes in contact with the cell, and from that point on, the piezo can pull it back, or piezo can be just holding there, and the cell generates the force to get to displacement X. That gives you the force. You trace the whole time sequence of the force. So let's calculate how much force can we measure. Can anybody see when I write this on the board? All of you can see? No? In the back? This might be trivial for some of you, those who are from mechanical engineering. Let's see how much force can we generate. So let's say, thanks. So if you have a beam, and let's say this beam is on the order of 1,000 micrometers, and the width, let's say 1 micrometer, and the depth is 3D, it looks like this. Okay, so that's the beam, and the depth here is, let's say, 10 micrometers. Okay? This is a long beam, 1 micron wide, 10 micron deep, and you can measure the force up or down. You measure because it's going to be formed like this. The same idea as AFM. So if this is made of single crystal silicon, okay, which is the microelectronic material that we can make beams out of it. Okay. Then, if I apply a force here, F, then the force is equals to the spring constant at the end, right? Spring constant at the end, so again K times delta, right? And we need to find out what is this K, how much force can we measure. Okay. So for this cantilever, this K is equals to 3EI over LQ. L is the length. Okay. E is the elastic constant of the material, elastic modulus of the material. So those who are biologists here, make sure that you get this point. That's the whole idea. Okay. It doesn't make sense, please stop me. So elastic modulus of the material tells you how soft or how hard the material is. I is the geometrical property. I in this case is 3E. I in this case is the thickness Q. So it will be T cube. Then the depth is D over 12. Okay. So this is the depth. D is the depth. T is the thickness. So T cube D over 12. So if T, as I said, is 1 micron, so this I is this T cube D divided by 12, right? That's the K. Okay. So for single crystal silicon, it depends which direction you have to make the beam, but without getting into too much detail, E is about 170 micronewton per micrometer square. Okay. And T is 1 micrometer in this case, D is 10 micrometer, and L, I said, is 1,000. Okay, 1,000 micrometer. So if you plug in the numbers, anybody has calculator? Computer? No? So if you calculate 3 times E, let's do this. So we have 3 times 170 times T is 1 cube, E is 10, divided by 12 times L is 1,000 cube, right? That's the calculation. Okay? So let's make it 12 instead of 10, or it's in the same, so it's the same order, right? So I can take 10 and 12 out. So you're getting some order of magnitude. So we have 3 times 17 times 10 to the power 4, right? And here divided by times 10 to the power, this is
Minus
10,
minus 9.
minus 9, right? That would be micronewton or micronewton. Oh, that's the, this is the spin constant, right? So this would be, so 3 times 17, so it's 45, about 50.
51.
Okay, so it's on the order of 50, right? Times 10 to the power minus 5 micronewton per micronewton. Okay? So how much force do I get? So this is 50 micronewton times minus 5. So this is 50 times 10 to the power minus 2 nanonewton per micronewton. Right? Because 1 nanonewton is 1,000th of a micronewton. So this is 10 to the power minus 5, this is minus 2. I convert from micronewton to nanonewton. Right? So if the spin deflects by 1 micron, I measure only 50. So this is 50 times 10 to the power 1 piconewton per micronewton. Right? Which is 500 piconewton per micronewton. So I'm measuring, if the beam deflects by about a micron, then I can pick up 500 piconewton force. Small. But not as small as we want. So if my camera or my measuring device can give displacements, if the measurement tool on the microscope can give you 0.1 micron resolution, right? If delta can be measured with 0.1 micrometer resolution, right? Then you get a force of 50 piconewton. That becomes your force level. So you can measure 50 piconewton force if you can measure 0.1 micron displacement in your microscope. So if you bring this system contact with the cell and the cell begins to pull it a little bit, and you see that the cantilever deforms by about 0.1 micron, you say, aha, the force that the cell is producing is 50 piconewton. Remember, what is the origin of the force? The origin is back to the molecular motors. A molecular motor produces on the order of 2 piconewton force. There could be hundreds of these molecular motors. At the beginning, when the cell comes down, there could be only few of these motors. You're generating on the order of 10 piconewton force. Right? If your cantilever can pick up 50 piconewton, it's not quite sufficient. But that gives you an idea. That's one of the springs. Now, if you put many, many springs, then you can increase the resolution. If you divide this spring constant, this is the spring constant, right? If you divide by, let's say, 10 of them are in series, then you can pick up 5 piconewton. That's the idea. Any question? Have you seen this kind of calculations before? No? High school? Okay. All right. So this sets the number into perspective, right? You can make something on the order of a millimeter in length, which is possible. Then you can pick up from looking at your microscope how much displacement is there. All right? Okay. Problem is not there. The problem is when you make something very, very soft, like if you K divided by N, very, very soft. That means the spring is very soft. It means the spring is linear but soft. Because small force can make it deform. Right? So what's the problem? So these springs are so soft that if I have one of them sitting here, and somebody just opens the door, just a little bit of air draft, you will see them deform. Okay? So we used to make this, and we used to see them deform, and I could not find out why they are bending. We close the door, and nobody is blowing anything, but we see them deforming. And we found out that every time I would move my leg, this would deform. Okay? The tiny bit of air circulation that is produced by moving my leg would deform this place. So that's the level of sensitivity that we have. Okay? What's the problem? The problem is the cells, if I want to measure forces on the cells, where do the cells live? What do the cells need to live? Media. In a medium, in a liquid medium, mostly aqueous medium, right? But the sensors, you have to make it in air, somewhere in the air, right? So, what's the problem?
And you get
Oh,
Other force.
what's that?
Other force.
Say it again?
I mean, other forces.
Other force. What other force?
Brownian movement, random movement in the media itself.
You are still in the air, right? You make the sensor very, very soft. You are still in the air. And this is a common problem for anything that you use in biological forces. So you better understand what I'm talking about. So you make something very soft in air. The cell is sitting in the water. So what do you have to do?
Plunge through the water.
Go from air to water, right? Seems very simple. So what's the problem? So here's an experiment I'm going to show now. And maybe that will eliminate some of the confusion. So what I have here is water, which is the case for the cell in point. And what I have here is a couple of silicon pieces. Okay? You can see silicon pieces here? Okay, here it is. Let's see if I have a white screen. No white screen. And this was the idea. There is a killer somewhere. So where is the killer? Let me show you. Now this is a non-trivial concept that I'm going to present now. And you might not have seen this before. So what I have here is silicon pieces, 2 silicon plates. Silicon is 2.5 times heavier than water. Okay? 2.5 times heavier than water. So if I take this out, and if I put some water there, and to contain this water, I'm still alive, right? This is water. So I put the water here. Okay. So if I take these plates now, which is 2.5 times heavier than water, what's going to happen?
It will sink.
It's going to drown, right?
Yeah.
That's what physics tells us, right? It's going to drown. So let's see what happens. Does it drown? Did we learn something wrong?
Surface tension.
Surface tension keeps it afloat. So how does it work? How does surface tension keep it afloat? Okay. So the way it works is so you have a plate. By the way, the silicon hydrophilic. Do you know what's hydrophilic? So the water tends to inundate it, right? It tends to fill. Why is it hydrophilic? Because well, silicon itself is not hydrophilic. I correct myself. Silicon produces a thin layer of oxide around it, silicon dioxide. Right? Silicon is metastable. So silicon itself cannot sustain in air. So immediately, as soon as it gets exposed to oxygen or air, it forms silicon oxide. Silicon oxide is glass which is hydrophilic. It wants to wet, right? So you would think that when I put this on water, the water wants to inundate it, fill it up, right? Because it wants to fill it up, it wants to cover. So there is no reason why it should float. In addition to becoming heavier, being heavier than water, right? So hydrophilic means if I take a glass surface and put a water droplet, it creates an angle called the contact angle. Contact angle phi is less than 90 degrees. That's what hydrophilicity means. Now, when phi is 0 degrees, perfectly wetting surface, how would the picture be when it's hydrophobic, like wax? the other way, right? If I put the same droplet on the wax, then it's going to look like this, just like when you have wax and water droplet. Like what you see when you walk out in the morning. On the leaves, right? Like lotus leaves in particular, they are highly hydrophobic because they have little beads on it, and those beads are coated by wax, and they make it hydrophobic. So it's called, that's how it can clean itself all the time. So that will be hydrophobic, but this is hydrophilic, this angle is less than 90 degrees. So how does this, so if I take the plate that is floating now, the plate is made of silicon dioxide because of the coating, right? So it should try to inundate the whole thing, but why it doesn't inundate? It doesn't inundate because if you think of it, this angle is very sacred. It's very, it has to be respected. So if you have the same surface, it always has to produce this angle. So if I'm taking this out of the water, you would see this forming a meniscus like this, right? You have seen this all the time. If you take something out of the water, it creates this meniscus like this all the time, right? Because it's hydrophilic, it wants to hold. It loves water, right? Hydrophilic. Now what happens when you try to inundate it, if you blow this corner up, this corner would look like this, right? Because I'm just blowing up the corner. The corner would look like this, right? So if you think of if I'm floating, then and if I have to respect this angle phi, how should I draw the meniscus? If I have to float, I'm assuming this is floating, and I have the proof in front of you. So why should I draw the meniscus? If this is where the water line is, where is the meniscus?
With respect to that curved edge of the.
Right, so the water is floating, right? So where is how does the water meet the plate?
In the middle. Right in the middle.
What's that?
Right in the middle of that curve. So the angle will be 0.
Like this?
No, it goes around.
It goes around?
No, it goes around the curve that you draw.
Goes something like this?
No, the contact angle will be 0. So,
Contact angle is 0?
yeah.
No, I'm already giving you a contact angle. Let's say 30 degrees.
Oh.
I just put it as a number.
Oh.
Right? There's only one way, one unique way this can happen. How can you respect the contact angle phi at the same time keep equilibrium? Well, imagine that I had this plate just contacting the water. Imagine the sequence of events in your mind. Okay? So if I have this contacting the water and I'm way up from the water level, then we know our everyday picture, which would be that the water line might be something like this. Okay? Now, as I keep moving, let's start the new page and blow this up. So, microscopically, I'm looking at the edge of the plate, right? So, if I'm going up, then my contact line may look like this. So this is the liquid, right? Does it make sense? No. Wake up, guys. It's wrong. So what's the contact angle here? This is the contact angle, right? But I told you already, the contact angle is phi, less than 90 degrees. So if the droplet looks like this, it should be like this. So I have to respect the contact angle. I cannot violate that one.
So then the water kind of hugs the surface.
Right.
You
It has
go
to
up.
go like this. Good. Just go like this. Right? I cannot violate the sanctity. So that would be the contact angle. Not this one. This is wrong. So this is the case when I'm pulling it up. Now I'm going down over time. I'm pushing it down. So the contact line is now beginning to move. Right? But every time it is respecting the contact angle. So when I'm here, maybe I'm going like this, vertical. And this becomes my contact angle. Right? If I'm moving down a little bit, let's say I'm over here. Now, this line and this line has to satisfy the contact angle, so I'm moving like this. Right? Keep thinking. When I'm here, maybe my contact line is like this. When I'm here, again, I have to satisfy the contact angle. Maybe it's like this now. When I keep going up, let's say I'm over here, my contact angle may look like this. This is my contact angle. Right? So I have come nearly to the top. My water surface is like here. So what is the force on this plate by the water? Gamma is the self-contact, is the surface energy. So gamma is the force that is holding it back. Right? If this plate is long, then the force that you get from the water surface, F is gamma, that is the force per unit length. So gamma is force per length, right? Which is like a curtain that you're holding it. Right? So it is the force on the curtain, force on the cloth that you're holding something. So that's gamma. Gamma is the force per unit length. Right? So the force that it is applying by the surface tension is just F equals gamma. And if it has already reached the top surface, then if this angle is, let's say, theta, then the vertical force, F vertical, will be how much? Vertical force is a component of the force, right? So that will be F times cosine theta. Right? That's the force it can provide, hold the plate. Right? That's what keeps this thing floating. Okay? So it gets some more help from where? To keep it afloat. Who else could be supporting it? Buoyancy. Right? Why buoyancy? Because it's underneath the water surface, right? Just like the boat goes underneath the outside water surface. So if the water surface is up there, so the buoyancy pushes it up again, right? Buoyancy pushes it up, but buoyancy is not enough because if it is only buoyancy, it will float, and it will inundate because it is much heavier than water. Right? But it is this guy that keeps the weight, keeps the float. Okay? So any time a solid, hydrophilic or hydrophobic, goes from air to water, it has to overcome this force, break this meniscus, after which the water will just float. Somebody have a question? Yes.
Now, does density play a role in how far is it?
Density, in terms of this force, no. Density comes over here. Right? Density comes when you want to find out what is the maximum size of this plate that can float. So if you have a plate that is long, to make calculations simple, so what will you do? You would say that the force per unit length of this plate would be F, the force would be gamma cosine theta, is a force that is pulling up, right? Plus the buoyancy force should be equal to the weight of the body to keep it afloat. Right? That's the maximum weight it can support. So, why, when we take a big piece of glass, like the silicon, it drowns? Why does it conflict with our understanding of the weight? Why does the bigger plate, bigger objects do not float? They're heavier in the water. Have you tried by jumping in the water and be afloat? No, it's not going to work. Capillarity is not enough. This is the effect of the size. Because this thing works along the perimeter. Right? So if you think of a circular plate. Think of a circular plate, then the capillary force would be something like pi 2 pi r times gamma, right? Because it's working along the perimeter. Gamma is per unit length, it's working along the perimeter. It's holding it up, right? And the weight of this plate, so weight stays as pi r squared, keeping the thickness the same, right? If the thickness is the same, it's called T. Keep the thickness. So if you keep making the plate larger and larger and larger, then what happens? So the weight goes as r squared, right? This is the weight as r squared. Capillarity remains linear. So capillarity would be, this is like gamma r, right? Gamma times r. If the size is small, this is r. If the size is small, then capillarity wins. You can float. If the size is large, the weight wins. You cannot float anymore. So what's the problem here? The problem is, if I want to measure the cell forces, things have to be small. I talked about micron size, right? Tiny, tiny size scale. Capillarity wins over gravity, over strength. So none of the sensors can survive. Going from air to water. Okay? That's the fundamental. No matter how you want to measure cell force, you want to make something very soft and inundate it and go to the water. It is this guy, the capillarity that kills you. Any other question on this basic idea? No? Okay, one more thing. So this is still floating, right? It's kind of interesting. Let me show you one other thing. And I will not discuss this if this experiment works. Whoa. Why is that? So if I took them away, how come? Why do they attract each other?
They want to minimize the surface energy.
They want to minimize the surface energy, right? So can you produce an example where they repel each other? Repel. They go away from one another. The soap will change the surface energy, right? But just by having the same material.
In the water?
What's that?
Once it's completely surrounded.
No. If it's completely in the water, then none of the capillary forces work anymore, right? You have to have the meniscus, right? Those meniscus has to be there. So you can create examples where they repel. You can create examples where they attract. More complicated issue. We won't get there. Okay, so we have to have these force sensors that has to be able to survive the meniscus. There are multiple ways of doing this. I think I will not discuss too much on this one. So this is to show how this meniscus works. See, this meniscus, how it goes out the way I was drawing on the board. Right? This is the meniscus that goes out. So basically, this force cannot break these soft sensors. It's the same way that water bugs can float and walk on water. And this is what I was talking about, the hydrophobic surface that makes the contact angle larger than 90 degrees. So there are lots of theories, I don't want to get into this, and one way to avoid it is to have these sensors covered by some other material. Okay, let's say in this case aluminum. So they are not, these beams that I talked about is very soft. It's soft when they're free, when you can move. But if you cover them by something else, then they are not free, meaning they're not soft, they're very rigid. So if you take one of these sensors and put it in the water with some etchant, in this case let's say some aluminum etchant, then the idea would be that this metal will be gone because this will be etched out, and then they'll be free, right? They'll be free. So when you take it out, then the water droplet would form around the sensor, and it will be always inundated. And then when you want to measure the cell force, you bring it back to the water, you form the meniscus again, and then inundate, and you contract the biological specimen. Basically, you don't make the meniscus ever see the sensor. So I'll show you a couple of examples here. So this is one of the sensors with many, many beams. parallel beams, beams in series. And this one, and this is the case where the sensor is sitting here. We're taking it out of the water, and it keeps the sensor inundated, and you'll see tiny droplet formed, and the sensor never got exposed to the air-water interface. This was taken at 5,000 frames per second. And this would be the case, I think, when this is the case when we are going back into the water, and if it plays, you will see it forms the lenses again. So it's just about to contact. And this is being supported by this droplet, just about to contact, and will form the contact. And now the sensor can go into the water. So in other words, the sensor never sees the lenses. Okay? Okay, I will skip this one. So should I continue or should I stop? I haven't talked about biology.
I think we need to hear a couple, like maybe introduce like biology.
Biology. Okay. So some examples of where the sensors can be used, right? Okay. So if you have questions on the sensors or capillarity, and I'll be here for 2 more minutes after the lecture, I'll be happy to talk. And if you have questions, there are a series of papers. If you look in the web page, you can take a look at them. Some theoretical, some experimental. So now that we know how to make the sensors, how to go from air to liquid, now we can do some cool experiments. One of the cool experiments is this experiment with neuroscience. Okay? So these are fruit flies. So you must have seen the fruit flies all the time when the fruit becomes rotten. So they are annoying, right? Tiny little flies. So this is the embryo of a fruit fly. And this is the central nervous system of the fruit fly embryo. And these are the axons. The axons are like long arm of a neuron cell that forms connection with another neuron cell, or in this case, a muscle. So these are called axons. The axons are, for us, very, very long. The axons should be 2 feet long. So it starts from somewhere near the backbone, it goes all the way to contact my muscles. And that's how I can move my muscles. So, what are we doing here? So the basic idea in this case is the neuron cells have the cell is the nucleus and the cell body, and then comes this long arm, the axon that I just showed you, and at the other end of the axon you form connection with another neuron cell or another muscle cell. You form a neuron-neuron junction that happens in our brain, or neuromuscular junction that happens to move our muscles. Okay? So they are called synapses. So the way it works is when there is a signal coming, the signal comes as a voltage pulse that runs along the long axon and comes to the end, and at the end, some funny things happen. The funny thing is, at the end, there are little vesicles. These vesicles contain neurotransmitters. Okay? Neurotransmitters. So, when the signal comes, electrical signal comes, and the voltage pulse comes very, very fast. Like, you know, the voltage runs through the electrical lines. The same thing with the axon, long axon that goes all the way to the next building. And these are the little vesicles that are waiting for the signal to come. So when the signal comes at the end, the vesicles fuse with the membrane of the axon and releases the contacts. These are the neurotransmitters. So when I want to move my arm, by the time I decide to move my arm, the signal is sent, okay, and the neurotransmitters are released. Like about to release the eye, it's released, right? And when this neurotransmitter is released to the muscle contracts. Remember, how does it contract? Back motors, right? The loop is closed. So, what happens with these vesicles? So if these vesicles were not there, I could not move my arm. If the vesicles would not be there, I could not memorize it. Right? So the basics of memory is rooted in this clustering of these neurotransmitter vesicles. So the more a synapse is used, the higher is the density of the vesicles at the end. So it remembers that it has been used before. That's the basis of memory. When you drive a car and you see a building next to the car, next to the road, you take a look at it, and those synapses that you have used has been used, and you can think about those buildings for a few moments, and then you begin to forget. If you see an accident on the road, a traumatic experience, then there's a huge release of these vesicles and a huge impulse that comes, and next time the vesicles that got released get replenished by new sets of vesicles. If you had originally 100 vesicles, and if you release 10 of them, how many do you have left? 90. And you would expect the synapse would recover back to 100, right? So if you have used this synapse many times, or if the synapse have been used to a traumatic experience, then instead of going back to 100, it goes back to, let's say, 110. Okay? So the fact that it goes back to 110 from 100 is like you are burning a CD. In case of neuron, you don't burn it, but you change the vesicle accumulation of the brain. So if it goes back from 100 to 110, that means next time the signal comes, next time the action potential comes, the voltage signal comes, you don't release 10. How many do you release? You release 11. This is called synaptic plasticity. Plasticity has gone up. Okay? So it remembers the usage of its past by changing its efficiency. Okay? Changing its efficiency. So this is the mechanism of memory. This is how Eric Kendall got Nobel Prize in 1992, right? By finding the pathway by which this vesicle accumulation goes up. Okay, so this is like the initial artistic rendition of vesicle accumulation at the end. And so the student who started this work, briefly shown here, so he takes this, in fact the student was Scott Sheehan, who gave the first talk, I think, right? He was a graduate student at that time. So he took this embryo, and so because they form neuromuscular junction at the end, so what do you expect at the end to see in terms of vesicles? There should be lots of vesicles, right? Because without the vesicles, this little guy cannot fly, right? So every nerve end would have lots of vesicles. So you would expect lots of vesicles at the end. So what Scott did is he said, well, people thought about this vesicle accumulation at the end is controlled by biochemical signal process when it's coming from the cell. So he wanted to ask this question, that is it really true? So what he did is he did this first experiment. He took this axon and cut it in the middle by laser beam, and then he measured how many vesicles are there at the end, and he found that the vesicle density has gone down. Okay, which is what is expected because it is disconnected from the cell, so there is no vesicle coming anymore. But then, for some reason which I still don't understand, he pulled this cut end by a micropipette. Okay, and all of you have seen micropipette in the labs by now. So he pulled it by a micropipette. And what he found is the vesicle density has gone up again. Okay? So he said, well, all we knew about neuroscience by that time is that once you cut it off, there should be no more vesicles because it's not getting any signal anymore. Right? But just by pulling it, he got them back again. So what does it tell you right away?
That there is a mechanical
There's a
aspect
force
to
that controls it. Right? There has to be some kind of force.
So
Yes.
do you the assumption being that the force is coming from the soma through the cytoskeleton?
Or the other way.
The
The
other
axon
way as well.
itself is producing the force. Remember what we discussed at the beginning. The cell itself produces the force, really.
Right, so
By
that's
creating
what I
one
meant.
force.
So I meant from the center, the soma, there is a
Correct,
force.
right. To the whole axon. So, if he can provide that force, he can restore the vesicle cell, right? Even though it is completely disconnected from the cell body, right? So the first hypothesis is axons must be an attention for neurotransmission, right? So then the question is, how do you know that these axons, before he cut it off, were under tension? So you need to measure the force. Right? That's where the MEMS force sensors come back. So then what we did, we took the same force sensors that I discussed so far, and so this is how it looks like. Remember the calculations we did on the board? We had 1 micron in dimension on the board, and about a millimeter length we chose, right? In our simple calculation. These are 2 millimeters long beams, and they're connected to this rigid arm, and at the end with a tiny plate that's going to attach to the axle and measure the force. Okay? So this is where we discuss the capillarity, right? This is why it has to break the meniscus and bring in contact with the axle. So this is the experiment. So it comes in contact with the axle, moves the axle. So this is the axle. This is the X displacement that we have been talking so far. See, if there was no force, these two would be in the same line. Now there is a gap between the two. So we measure the force displacement, and we find that it does not go to the origin. So there is a force that the axon has even before it touches the axon. So there's another evidence that there is a force. Every time we touch the axon, it produces a huge gap. So it's like taking a rope which was under tension, you cut the rope springs, right? So this is another visual demonstration that the axon was under tension. So basically what I, let me just show you one more thing, is that it looks like the axons are under tension, and if you take the tension out, they cannot have the vesicles at the end. And it turns out that if you can increase the tension mechanically by the same ropes, the vesicle density goes up. So by simply changing the tension alone, you can control how much vesicle you can have at the end. So where is the force coming from again? The force comes from the axon itself. Right? And by increasing the force or reducing the force, the axon might be modulating this vesicle density.
So when you're doing this, when you're pulling it with the pipette, the broken axon, you assume that, again, it's going to be the cytoskeletal elements like the motors that are transferring that force, which is causing the vesicles to move back.
Right.
So have you, while doing the pulling, tried to inhibit the action of one of those cytoskeletal elements to see which is the one that is stimulated?
Yeah, we did that too.
Okay.
Yeah. So what you find is if you take out the actin filaments, okay, then it perturbs the vesicle accumulation. Yeah. But there are two fundamental questions. Who creates this force in the axon? This is a very basic question. Which means that if I think about the animal world, who has these neurons attached to it? The question is it universal that all axons are under tension? Yeah. Who creates this force? And the second is how does that force control the vesicle accumulation? Two basic questions. So I'm sorry I took only 10 minutes to talk about biology, but I thought once you go through some calculations, it will help to think about sensors, how you calculate the force, how you measure the surface, and so on.
Did they all pass?
I'm going to take the test now.
Is there any association between the length of the axon and the tension force?
Good question. We don't know.
Can you repeat the question?
The question is there any correlation between the length of the axon and the force? We don't know. The only animal that we have studied is the fruit flies, embryos. But when the fruit fly grows, then it becomes longer and longer. And whether the force is increased or not, we don't know. My belief is probably not. Probably not. It may be related to the diameter of the axon, not so much to the length of the axon. More diameter, more cytoskeleton, more motor proteins, not so much of the length.
But a related question is, along the axon, there must be a force distribution.
Right,
What you're measuring is right at the
ends.
Right.
Correct. So there could be different amounts of motor proteins getting engaged
Yeah,
along the
in
length.
vivo, it's actually interacting with the surrounding.
All together, right. This is all kind of hardwired network. All together.
Well, let me take a minute to, let's thank Taher again, and I got to present you also with the lovely plaque. And we did have some time for discussion. Lunch will be appearing in about 12 minutes outside. So do you have questions? More questions? I don't know. What is it?
Yeah, there is still time, there is still time for discussion and so on and so forth. Lunch is perfect.
All right. Yes, go ahead. How do you detect the cell force? Under the water? Yeah, so the question is, is it possible to measure the force inside the cell, right? Because then you have to get into the cell, you perturb the cell significantly. People have measured the force inside the cell using magnetic beads. So this was a work done about 15 years ago in Germany. So they put the beads. So in that case, the way to do it is like this. This is the work done in Munich. So you put the cell and you take the magnetic bead and you poke the bead with some appropriate molecules that the cells like. So you put the bead gets attached here, and as soon the bead gets engulfed inside, right? And then the bead eventually gets into the cell with the coating, and it eventually gets bound to some cytoskeleton inside. And you can see the bead because they fluoresce. They have some light coming out. And then you apply an external magnetic field. So if you bring a magnet, this is north south, and the bead was magnetic. So then you can apply the force in the bead, and you see, because you know how much magnetic field you're producing. So then by looking at how much the bead moves, you know how much the force on the sensor. But not using an external force sensor that goes in.
But people have also measured the force by putting DNA.
Yeah, that's external. Yeah. Hard question is, can you get into the cell while it is living, it's doing its activity?
Actually,
Yes, go
I
ahead.
had a comment about this experiment. So I'm a nanoparticle person. I think later in the course you're going to see someone talk about nanotoxicity. Is that right? That sounds
Yes.
useful.
Yes.
Because, you know, okay, people who are interested in particles getting into cells and see all kinds of issues with
Absolutely. Yeah.
this part happening at all. And,
Right.
you know,
That's right.
right? In terms of distribution, effect on the cell. You know, so yeah, maybe it's gotten in there, but I
Yeah.
don't really know what's attached to it. Not really.
Yeah, that's right. And you cannot control just one bead. There could be hundreds of them getting in, right? So all of them are creating things inside the cell. Yeah, so toxicity is a big issue. Toxicity in general terms, because once they get in, the cells may start doing something. Right? That's general. So what you measure is not what the original cell will do. What you measure is because you're measuring it.
So I guess, you know, if we try to imagine what kind of cool thing can we do inside a cell, let's try to imagine those. Let's try to imagine those.
Yeah.
Do we have, you know, a molecule that would be in a cell normally that would have some change in configuration naturally as a function of some force? And could we try to do a single molecule experiment in a cell? Okay, so that kind of thing could be a frontier area that maybe we should think about, see what we can figure out. If you can measure with enough resolution in the experiment, right? Because if you're going to do anything optical, you got about a 2 nanometer limit, right? Unless you do something fancy.
Yeah, what people have been able to do is using laser to cut fibers inside the cell and see the contraction of the fiber. That
Yeah, approach, isn't it? I also wanted to make a comment, as long as we're sitting here, that the idea of the meniscus is also very important in people who want to do patterning of surfaces. Like you may have heard of 10 nanolithography, right? We have an AFX tip with molecules and you put things on a surface, right, to make a nice pattern surface. Well, it turns out in the original experiments the people did, they found it worked really well on days like today when it's very humid, and it didn't work so well for winter when things were very dry. And they eventually figured out that you really need a water meniscus between your tip and your surface to help transport your molecules, even if they're kind of hydrophobic, from the tip to the surface. So now they do everything in controlled humidity chambers. So again, these kind of ideas that go back to the principles we're all very familiar with are very important on very submicron scales.
Change the humidity, basically you change the surface energy, the gamma, you just change it here, and in addition to other things.
Okay, do we have other comments or questions? Yes?
So for doing this type of measurement, you've got the reaction.
No, not anymore. So the first experiment when you cut the axis, that is when you wanted to find out if you disconnect the synapse, the endpoint in the cell, what happens. But when you want to measure the force, no. You want to keep the axon as it is and then it will keep it alive. So the way we do it is it's like a surgery. Cut the embryo a little bit and then probe goes in and then plucks the axon and then let it go. And in the plucking process, you measure the force and you leave it alone. So like when you want to measure the force in the brain, the neurosurgeon will go in, pluck one axon, pull it, let it go, and then seal it back. Excuse me.
When you talk about the AFM, one of the applications of AFM is to measure interactive force in molecular level, right? So if we are trying to measure the interaction force between specific ligands and receptors on the cell membrane, so can we just call the coating the ligand molecules on the AFM tip and then close to the cell membrane? And then measure the force, right?
Yes, yes.
So, but how do we know the force that we measure is one molecule to the one set of receptor or a bunch of the molecules to a few receptors?
Right, good question. So there are two ways of doing this. One is if you reduce the density of the receptors, or the ligands in this case, with the tip, so that the likelihood of having too many is very few. So if you make the coatings very, very low density coating, so that given the surface area of the FM tip, you have most likely 1, 2, 3, something like that, discrete number. And then you form the receptor ligand binding, and then as you pull it out, let's say you have 3 of them, and then you have one by one breaking. So you have a discrete force drop. And from a discrete force drop, you can say, okay, I have broken one by one. That gives you the force for each one. And that has been used before. The argument has been pushed that if I get only one jump and then I'm off, then I must have only one. So by changing the density to very, very low level, you make the number of molecules just a few.
So we can control the intensity.
Right.
Thank you.
So that's sort of like the Millikan oil drop experiment, where you try to get the charge on an electron, and differences that were sort of discrete intervals.
Right.
You can kind of figure out that okay, that must be even if you don't have one electron in the drop of oil.
Correct.
Right.
Classical experiment.
Have you ever tried to use this as to measure the protein and protein interaction force? Because maybe when we use this system, maybe we can select some antibody to the energy to select the best affinity to the to select the best affinity of antibody to the some energy by using the pattern. Do you have any try to
You see,
try
the,
to vertically sanction force?
yeah, you can do that in principle, but you'd rather go to AFM to do that, because AFM gives you very fine resolution. And if your force measurement range is few piconewtons, then you don't need these MEMS devices. So this is more meant to contact larger surface area of the cell. So to create the focal addition complex that I described at the beginning, we need on the order of a micron or more surface area. So it's not that very small. So these are more appropriate when you want to measure slightly larger scale. If you want to measure receptor ligand type of force, then you don't need large surface area. In fact, you want smaller and smaller surface area. So in that case, AFM is the best.
Other comments or questions? If not, let's thank Professor Saif again.